Hello everyone. This tutorial is for the Bio 105 Exercise for Sampling Macro-invertebrates, one of my personal favorite labs. So some of the objectives in this lab are to demonstrate how to sample an aquatic ecosystem using basic equipment and techniques, to identify aquatic and macro-invertebrates using a dichotomous key and field guides, how to use a biotic index to make assumptions about the quality of water in an aquatic ecosystem, and explain how the diversity of macro-invertebrates can be used to assess water quality. A few relevant terms: a biotic index is a mathematical assessment of an environment based on the type and relative numbers of organisms present. This is similar to our Simpson's diversity index that we calculated before, except this one is a weighted index, and we will talk about why that is. Macro-invertebrate is any invertebrate, an animal without a backbone, large enough to be seen without magnification. So here we've got three different insects, but this also includes things like snails and crustaceans. So a biotic index is a way to monitor a lake, pond, any waterway by sampling the organisms that are present. It's based on the fact that different invertebrates differ in their ability to tolerate pollution. Meaning that if you see a lot of organisms that, like this mayfly here, are not very tolerant to pollution, that means you've got a good water quality. But if you see a lot of organisms that are tolerant to pollution, you might have a bad water quality. Nice thing about biotic indexes, they reflect what's happened over time. So what can happen is you can have a pollution event that sort of gets washed downstream, and if you sampled a few days later, you wouldn't notice it. But the invertebrates in that aquatic community would still be affected by what happened. Another nice thing about it is it uses simple equipment, and it can be performed by citizen scientists and volunteers. So a lot of times these are used when folks want to volunteer to help monitor water quality. The equipment is simple, and they can help out by gathering data for scientists. So let's run through an example. First, let's talk about a few of the cast of characters. This one is a mayfly larva. They have very low resistance to pollution or low oxygen levels. And so in our index, we class them as a 4, which means they have a high score of 4. And we'll talk about what that means in just a minute. But their presence tends to indicate low or no levels of pollution. Opposite end of the spectrum, we've got the rat-tailed maggot. They're very resistant to pollution and low oxygen. They are a class 1 organism that gives them an index score of 1. So their presence indicates high levels of pollution or very low oxygen. So these guys have been found in sewage pretreatment lagoons. That's how tough they are. I once took a aquatic insect identification class, and we had found a few of these. Most other insects, you can put them in ethanol and they die within seconds and will be preserved. These guys, we had to drop them in formalin, which is basically formaldehyde, and they would still persist for like a half an hour. They're pretty tough. And these guys are going to be super common. You'll probably see a bunch of them. They're dragonfly larvae. They're sort of in the middle as far as their resistance to pollution and low oxygen. So they have a class of 3 and an index score of 3. So their presence means somewhere moderate or low levels of pollution. And the interesting thing about these guys, besides the fact that they have mouth parts that'll shoot out and grab other insects and eat them, but they also will swim around. They'll scoot around by basically forcing water out of their butt. All right, so let's do an index. Let's say we went out, we took a sample from a pond, and we counted up all the organisms that were in there. So what we would do is we would identify the species of insect and count up how many was there. So in this case, we had 75 mayflies, 20 dragonflies, and 5 of those rat-tailed maggots. The index score that we talked about, so it is weighted. It is weighted towards the insects that are least resistant to pollution. Right? So their presence means that you've got a healthy pond. So our mayflies, every mayfly that you find has a score of 4. So for every one, so we had 75 of them, so we would multiply times 4. That gives us 300 as a score for all of our dragonflies. all of our mayflies. Our dragonflies, there's 20 of them. Their score is 3, so they contribute 60. So then if we look at our rat-tailed maggots, there was 5 of those. Their score is only 1, and so they contribute 5. So we sum up all of those n times a's. That gives us 365. We then divide that by n, which is our total number of organisms. In this case, we made it easy in that it is 100. And that gives us our biotic index. In this case, 3.65. So if we look over here, that tells us that our health of our little pond here is excellent. Right? Anything over 3.6 is very good health. We can contrast that with our next sample, where we have 5 mayflies, 20 dragonflies, and 75 of those rat-tailed maggots. Again, we multiply times our index score, and then add up all of our n times a's, and divide it by the total number of organisms we found, which was, again, 100. And so we get a score, in this case, of 1.55. So we look down here, anything less than, anything between 1 and 2.0 is going to be poor. Right? So that indicates that there's something wrong with this particular ecosystem. All right, there's something going on. There's low oxygen. There's pollution input. We don't know what it is, but it tells us that there's something not right here. So, on the day of lab, the folks of you who are in room 241 are going to meet over here by the corner of the blue field. This is out behind Secaucus dormitory. And the folks that are in room 239 are going to meet over on the other side of the pond, between the pond and the jogging track. We're going to be outside the lab, so make sure but you still need to make sure you're going to wear closed shoes, long pants. We're going to be outside the whole lab, rain or shine, so dress appropriately. Sunscreen and insect repellent are recommended, but you need to bring your own. It's going to be wet and muddy, so especially the folks that are going to be taking samples, they're going to be down by the edge of the pond. If you've got rubber boots, they are recommended. You're going to be wearing a mask and gloves for the entire lab, and we're going to maintain our 6-foot distancing. One student in each group is going to pull a sample from the pond. We're going to put it in a bucket, and then each student is going to pull from this sample and sort the invertebrates in their individual trays. Each group is going to identify 100 individual invertebrates, and then each group is going to consolidate their data and calculate their biotic index. Then you're going to use that data to answer the lab questions. So thank you for listening. We'll see you in lab.